OmicsLogic programs. Over the course of the past couple of years, we have been developing a program that consists of a few elements that have been tested extensively by our partners in various institutions in the US and around the world. We have been working with a number of institutions that have been offering these materials to their graduate and undergraduate students, as well as high school students. And we've seen a lot of success in terms of students from different backgrounds, including non-engineering backgrounds, being able to learn about bioinformatics as well as apply bioinformatics to their research projects, either by themselves independently or working as a group and with their faculty. For those of you that are just joining, if you have trouble hearing the voice or you have trouble seeing the slides, we will have a recorded version of this presentation that we will be putting on our social media on YouTube, and you'll be able to see it as well. Importantly, the program is developed in collaboration with top faculty at universities both in the US and around the world. Our major partner in developing the materials and the learning experience that we are offering you is with the Tauber Bioinformatics Research Center. The Research Center is located at University of Haifa in Israel, and it has been working on development of a multi-omics integration big data analytics platform called the T-BioInfo Platform. First, let me briefly introduce myself. My name is Ilya Brodsky. I'm the CEO and co-founder of Pine Biotech. And our mission at Pine Biotech is to harness big data to allow researchers to leverage effective tools with logic and understanding to make discoveries using high throughput omics data. So what we do in our company is develop solutions for big data analysis and help apply those solutions in an effective way by offering training and support to research teams in top universities. I am privileged to work with a team that includes our founders, Dr. Alfred Tauber, who's an internationally recognized physician with a background in immunology, and Dr. Leonid Brodsky, who is a well-recognized leader in the field of bioinformatics with hundreds of publications on development of algorithmic approaches to analysis of omics data, as well as biostatistician and mathematician, including our team that is working to develop and support the platform, as well as the educational resources that we're going to be offering you as a part of this program. Julia Panov is a PhD student and a bioinformatician at University of Haifa in Israel with a specialization in neurodegenerative diseases. Avi Titievsky, who is a computer scientist working on the development and support of the T-BioInfo high-performance computing platform. Dr. Mahit Mazumdar, who is a computational biologist and will be working closely with you in this program to help you develop research projects and make sure that you have all the relevant materials and can succeed in the program. Joshua McKendall and Suraj Patel, who are working on developing the resources that we'll be using, including the educational portal and the coding platform, and Bipsa Biswas, who I'm sure many of you are already familiar with. She is our bioinformatics community manager, and she will be interacting with you to make sure that all of the logistics of the program are clear and you have access to everything that we will be using throughout this program. Now, we also have a trainer with us. She has a PhD in bioinformatics from the Bioinformatics Center, CSIR Institute of Microbial Technology in Chandigarh, India, as well as from JNU in New Delhi in India. Her PhD is focused on the domain of cancer genomics, specifically for liver cancer. So Dr. Harpreet Kaur will be assisting with conducting the sessions in this program, as well as helping you understand and clarify any kinds of applications of bioinformatics to the different project examples. She has experience in both using big data analysis tools, incorporating big data into usable interfaces and databases, has experience in Python and R, and will be really key for those of you that want to understand how bioinformatics is used in big data projects. So Harpreet, I would like to pass it on to you now so that you can maybe tell us a little bit more about your background.
Okay. Thank you, Ilya. So let me share my screen. Okay, thank you. So thank you, Ilya. Everybody, myself, Harpreet, as already Ilya introduced me. And just look at just my educational background. I have done my PhD in bioinformatics, and I have background, especially my post-graduation in molecular biology and biochemistry, and I have done my graduation in biotechnology. So in my research interest, I have mainly focuses cancer genomics and bioinformatics, machine learning, biomarker discovery, and various data resource development on different aspects of the human health. So I have nearly 14 research publications with nearly 200 Google Scholar citations, and I got various awards from European Association for the Study of the Liver as Young Investigator Oversee Award. And then I have got Institute of from the Scholar as Young Researcher Awards, and I have got my scholarship from the CSIR, that is Council of Scientific and Industrial Research. And this is what I have done my PhD. So my PhD is mainly focused on the topic that is computer-aided identification of genetic biomarker for the predicting liver cancer and its prognosis, where apart from exploring the genomic data or omics data, we have implemented artificial intelligence approaches, and we have where we have applied machine learning plus survival analysis approach and various statistical analysis approaches on three main omics layers, that is RNA or the transcriptome layer, then transcriptome and methylation layer, that is epigenetic, and then RNA, methylation, and miRNA. So first of all, we have developed a resource that is called Cancer Liver. It is kind of a knowledge base where we have integrated information related to already existing biomarkers, that is 594 biomarkers that are associated with the liver cancer, and we have processed data sets that are available in the all different platforms using a uniform pipeline for nearly 10,000 samples, and we ultimately integrated in form of a web server called a Cancer Liver, and it is freely available. In the second work, where I have focused on identifying the SCC and discriminating SCC from the normal samples, and here we have mainly explored our RNA data of the patients, and we have identified three biomarkers that include CLAC1B, PRC, and FCN3 to classify HCC samples from the normal samples. Here we have nearly used more than 4,000 samples. And in the second part, where we have tried to classify the second form of the liver cancer, that is cholangiocarcinoma from HCC and the normal samples, and we have identified three genes that is LAM-C2, ITZA, and C2 as CCA specific signatures. In the third part, we have tried to classify early and the late stage samples of the SCC patients. Here we have explored RNA and the methylation data, and we have identified 51 features that include 30 RNA transcripts and 21 methylation CpG sites to classify early and the late stage samples of the SCC. In the last part, we have tried to predict the reoccurrence risk groups using 3 omics layers, that is RNA, MIR, and methylation, using survival analysis, and we have identified 90 RNA features, 50 miRNA features, and 50 methylation features that can stratify high and low risk of the reoccurrence risk of SCC patients. So let me just explain only 2 topics. So first of all, how we process these data sets and from where we have downloaded these data sets for all these studies. So here the 2 major public repositories, that is GEO and the GDC data portal, from where we have downloaded clinical as well as omics data set using various packages. Like from the GEO, we have used GEO query package, and from GDC data portal, we have used GDC client package. And once we have obtained the raw data sets, then we have used various pre-processing packages to process these data sets, like Affymetrix is processed using oligo package from the bioconductor, Illumina and Azilent using Lemma and Lumi package, and high throughput sequencing is followed process that is followed by TCGA datasets. So finally, we compile these datasets and clinical information in form of matrix, where sample is present in the columns and genes are present in the rows. So to further process these data sets, we have applied various statistical approaches and normalization approaches. And finally, we have performed quantile normalization to reduce any cross-platform artifacts using pre-processed core packages. And then by applying machine learning on the first project, we have 30 data sets, and we have divided these data sets into 3 categories: training and differential gene expression analysis, and 2 external 4 data sets we kept for the external validation cohort. So first of all, on 27 data sets that are kept for differential expression analysis, which contain nearly 2000 samples, we have performed differential gene expression analysis, and we scrutinized 26 genes that were found to be overlapping between SCC and the non-tumor samples. And once we have collected these 26 genes, then on the training data set, we have tried to reduce these genes in such a way that the performance shouldn't be reduced. We should obtain a particular signature set that must have minimum number of the features. So we have applied various feature reduction approaches. So we finally come up with the 3 genes, that is FCN3, CLAC1, and PRC1. And then we have developed machine learning prediction models. We have explored various machine learning classifiers here, like logistic regression, SVM, RVF, LDA, ridge classifier, and developed various machine learning prediction models. And finally, performance of these models is validated and evaluated on 4 external validation cohorts, which contain at least more than 1,500 samples. And eventually, we have observed that these gene prediction model based on this 3 gene signature set classifies HCC and the normal samples with sensitivity, specificity, accuracy nearly 95%. Apart from scrutinizing its diagnostic potential, we have also analyzed its prognostic potential. That is, whether these genes can also predict survival risk groups of SCC patients. So here we have considered 2 data sets, one is TCG and GSC, and then we have performed univariate survival analysis using 2 types of survival data, that is overall survival and disease-free survival or reoccurrence-free survival. And finally, we found that these 3 genes independently stratify survival risk groups of the patients with the p-value less than 0.05 and hazard risk at least 1.5. And eventually these prediction models are integrated in form of a web server, that is SCC PRED. And this paper is published in January, in Frontier in Genetics in the January 2020. In the second project, we have tried to classify early and the late stage samples based on two types of data. Here we have explored RNA expression data and methylation data. So we have divided these data sets into early and the late stage samples. And then we have divided these data sets based on training and validation cohorts. So to reduce, as the number of the features is very large, that is 60,000 RNA transcripts and nearly 5 lakh CpG sites. So we have applied first differential expression analysis approach and differential methylation, and we found nearly 103 RNA transcript and 103 RNA transcript and nearly 450 methylation CpG sites. To further reduce features from this, we have applied various feature selection approaches, that is K-based, FCBF, simple threshold based. So finally, we obtained 30 RNA transcript and 21 CpG sites. And then prediction model based, we have developed various machine learning prediction model, and we have observed the prediction model based on them able to classify early and the late stage sample with more than 75% accuracy. Then we try to combine them and performance is improved to nearly 79%. So this finally all the prediction models are integrated in form of a web server, that is Cancer LSP. And this paper is published in January 2019 in PLOS One. And here my main focus is how I can help you, any participants, in understanding the bioinformatics application through lectures, tutorials, or any kind of research projects. So I'm happy to help you, all the research interns in their research projects, their proposal and analysis designs. Thank you.
Thank you very much, Harpreet. And for all of you that are interested to learn more about Harpreet's background, as well as her research projects, you're welcome to look her up on the edu.t-bio.info portal. She has a profile and has gone through the materials that we are sharing with you extensively. And we hope that this program is going to be effective for you as well as it was for her. So what are we going to learn in OmicsLogic Transcriptomics? In this program, we will see how, relying on analytical methods and reference databases, researchers can study RNA-seq data to understand how gene expression variation is studied across many different conditions. To do so effectively, bioinformaticians leverage big data sets, processing methods organized into bioinformatics pipelines, and perform exploratory data analysis, normalization, statistical analysis, and rigorous hypothesis testing. So in this program, we will cover all aspects of this process, from exploring the data to making sure that the data is ready for analysis, and then learning how to train and understand the validation and the accuracy of such machine learning methods as you've seen today. We will also discuss the future of RNA-seq and how RNA-seq analysis has been changing over time because of the changing technologies, including single-cell RNA-seq. Next-generation sequencing allows us to study data at the subcellular level. So whenever we talk about phenotype, we're typically talking about a description of an external characteristic of a sample. As we go deeper, we can understand the variation at the genomic level, study epigenomic variation, and what we are talking about today is the analysis of expression data. Expression data primarily focuses on two aspects. The first one is gene and isoform expression, and because genes are typically not produced in the exact same way across all conditions and patients, or organisms, we're also going to be talking about alternative splicing. So when we talk about mRNA, we're actually discussing the process of translating the code that is in the DNA before it becomes a protein. For example, when we speak about HER2 as a protein that is being expressed and used for diagnostic purposes, we can capture the mRNA that encodes for the HER2 protein and understand how it is produced, in what quantities, as well as what types of variations we can find from what is regularly been noticed in healthy samples. So when we talk about expression of mRNA and alternative splicing of mRNA, we can explore and annotate the quantities and the variation of specific messenger RNA that are likely to become either complete, fully functioning, or potentially alternatively functioning proteins that are used for a variety of purposes. When we previously used microarray technologies to study mRNA expression, this was done using hybridization, which is unfortunately not as detailed as next generation sequencing for RNA-seq. So in this program, we will primarily focus on the expression analysis using next generation sequencing, using such platforms as the Illumina platform, which have a broader dynamic range, allow us to study both the quantities and the variation at the isoform level. In many cases, isoforms have been shown to have an additional informative component to distinguish between different types of conditions, an important aspect that we will highlight in the beginning of this program. Ultimately, when we turn to bioinformatics, our goal is to find associations between various types of data, including the association between clinical data and omics data, which allows us to understand how different diseases that we describe in clinical terms are represented at the subcellular level, at the molecular level, and potentially how this can change the way that diagnostics and therapeutics are being used in precision medicine. But in the program, we will also cover other projects that are not related to medical applications. For the biomedical projects using RNA-seq data, we will study such topics as diagnostics of tumor biology, including the differences between the tumor and the microenvironment. We will look at disease classification and patient stratification, which is something that Harpreet alluded to in her presentation, as well as look at single cell and tissue composition, important new frontiers being opened up by single cell RNA sequencing. So overall, in this program, we will have 6 different sessions. In our first session today, we are going to review the tools and resources that we will use throughout this program and how they will build up the skills and the important background understanding for all of the participants to start working on independent projects. Then we will focus on processing pipelines that combine multiple scripts and algorithms, including pre-processing, mapping, and quantification to produce a table of expression. Then we will turn to the analysis, exploration, normalization, and visualization of individual genes and isoforms, as well as bulk understanding of data so that we can explore patterns and understand what is in the data that we have processed. Then we will turn to the second part of the program, when we will talk about big data mining, machine learning, and predictive analysis, introduce single-cell RNA-seq data, and turn to hands-on project examples, where we will cover important examples in cancer, infectious diseases, and different types of biotechnology applications. When you have registered, you probably saw that we provide a detailed schedule for the program. After you complete the registration, the next step is to join the OmicsLogic program, which is a section on the educational portal that we have where all of these materials are clearly explained and combined together into a syllabus for the program. So to talk a little bit about the program logistics and how they are organized, I'm going to pass this on to Bipsa Biswas.
Thank you, Ilya. I hope everyone can see my screen.
Yes, we can see it.
Okay, thank you. So I will start with how the registration process works for those of you who are joining us for the first time or are yet to register. So this is the registration page. When you come on this page, as Ilya has been showing, you see we have all of the sessions listed down, the different dates of the program and the topics that we will be covering for the program. So if you go down, you will be finding that there are some reviews from participants who had been part of OmicsLogic programs previously. Then you can look at the courses which are available. So to register, all you have to do is if you click on the pricing option, you will see that there are 3 options that you can register with, and they all come with different prices, and they all have different features associated with them. And just to quickly go over these features, so the basic membership is something that everyone has access to, and you will have as part of this program. For the intermediate membership, any participant who opts for this membership will also get access to one-on-one mentor sessions if they require assistance during the entire length of the next 1 month. And for the advanced courses, especially for participants who are doing a research project and need more guidance and more time, we'll be giving them an extension of 1 more month where they can actually go ahead and work with our own group of research interns who will be also working closely on projects and also with our mentors. So once you see here, if you select each of these options, you can go ahead and complete your registration. So once your registration is done, you come to this page. So this is one of the resources that all of you have access to. And this is the educational organization page. So we treat the organization page as what you would typically treat a classroom as, like a lot of people learning together in one place. So you can call it a virtual classroom. So you see here we have different tabs, which I'm coming to very soon. You see the program description here. You do not need to register for all events if you're already registered for the program. And you can see that today we are here on 15th September. So if you go around, you can see all the different dates the program is going to be. Here you can see all the other participants in the program who are doing the different courses and what they are exploring. So you can actually connect with another person as part of the program and work together. And there are different tabs. So we are going to be coming to our faculty. So if you go down here right in the menu, you have a few more resources, faculty and syllabus that you can go through. Then you have the forum. So the forum would be a main point of conversation and communication between you and us. So if you have any questions, you can actually put them down in the forum. There are already a few topics that you can create and you can also post about. Every day, the session recordings will be posted here. So in case you do not find the email, you can again come back to the forum and check the session recording. All participants will have also the links to these, which I have already emailed to them. And if you have not received it, you can again reach out to me. And also we feature top performers every week on the basis of the asynchronous courses which they do. And these are a few of the asynchronous courses that you will have access to. So each of these courses come with their own certificates, which are different from the entire OmicsLogic Transcriptomics program certificate. So you can earn both in addition to the OmicsLogic program certificate. And if you look here, there are also the members of the program. You can go to everybody's profile and have a look at what the other person is doing. There are also some asynchronous projects which are associated and given as part of this program. And you will be able to explore these projects like you do the asynchronous courses. So here is also another tab, which is the progress tab. So the progress tab shows your progress as a group, as a whole, and you will be able to see. So right now we have just started, so there is not much progress. As we go through the different weeks, you will be able to see the performance of the entire group here, and also the different points that you will be earning on the education portal. So the points you earn on the education portal are by doing the courses, and I'm also going to be demonstrating them very soon. And there are also points for the server access. So if anyone else who has not received the server access, you'll be getting them soon today. So each of these adds to the points and also adds to the entire course certificate that you would be getting. So we offer two kinds of certificates. One is a certificate of excellence and a certificate of participation. Any participant who has over 10,000 points is awarded the certificate of excellence, and also anybody who does a project. So going forward, I just wanted to introduce everybody to what a profile looks like. Now, you all have profiles when you join the program. So I'm just using Connor's profile as an example here. So you can see that Connor is already registered. He has all of his social media links. If you're a researcher and you have an ORCID ID, you can actually add that, or you can add any of your social media links. We would recommend that you definitely add your LinkedIn ID, because a lot of our people collaborate with each other within the community, and they would be reaching out to you as well. So, and you can see the points that Connor has collected after doing a few of the courses that he has completed. So Connor has already been a part of a different program with us, and you can see that his certificate is also there from a previous program. So Connor has updated his profile completely. Yes, and just going back to the chat. So Siddhant, yes, Siddhant, you will have access to all of these courses. Right. So how to edit your profile. So a lot of you will see that your profile is not updated, meaning that your name is probably a username. You will have to update your full name, because this will be displayed in the course certificates that you will be getting here. So you can go just to profile and the edit option, and you will be able to edit all of these fields right here. So, and moving on, I just wanted to show the course certificates and the courses. So here you will see the courses that you are taking or you are enrolled in. So if you go to certificates from here, you will be able to see that the different certificates that you have got for the different programs, right? So right now Connor has no certificate. So moving on, there are also two other platforms that you will have access to. This is our code OmicsLogic platform, which our mentors would be showing you in a few more minutes. And the other is the server T-BioInfo platform. So with this, I'm going to be stopping my screen share. Ilya, if you can please take over. Thank you so much.
Thank you, Beepsa. And it's great to see all of the questions. We will definitely be covering many of these topics as we go on. So as Beepsa mentioned, your profile is the center where you will see all of your activity. You will be able to also see the different types of activity, like pipelines or completing your lessons, or even running some exercise on the coding platform. All of it is going to be integrated using our point system, which gives both us and you an indication of where you are, excuse me, compared to other participants of the program. Each course has a number of quizzes, and you will be able to review those and try them several different times. You will also see your progress over time so that you can evaluate how much effort goes into completing the individual exercises and assignments. And at the end, as Beepsa mentioned, you will receive a certificate as well as certificate for completing courses, and an overall certificate for the full program. So let's review several different elements that this program relies on. One of them is interactive sessions, just like we're having today. The goal of interactive sessions is to provide a live review of the topics and give you some practical exercises that you can do together with a guided presentation by a mentor. In between the sessions, you can rely on self-guided study materials to complete asynchronous courses, work on projects, practice coding exercises, and running pipelines. And then the practical assignments are going to be given every several sessions so that you have enough time to practice and work on your own. If there are any technical challenges, those will be reviewed in the interactive sessions. The coursework that we've prepared for this program includes Transcriptomics 1, 2, 3, and 4. You can see it on this link, edu.t-bio.info collections transcriptomics, or under the program, which is under the courses tab. Each course has several elements that you will see as you go to that course page. It starts off with an introductory video. And inside the course, you will have several different additional resources that you can benefit from. There are different topics that are included and keywords underlined. You can also download the full course material as a PDF. There's a lesson forum where you can ask questions and get answers. And you can also manipulate the font size and images that you can zoom in or increase the font size. So inside the course, you will see that there are both technical explanations, again with key terms that are highlighted that you can learn from if you're not familiar with those words. And it's important to remember that we try to prepare these materials for both people that do have a background in cell and molecular biology and bioinformatics, and those that do not. So we're trying to really make it easy for anyone to jump in and benefit the most from the materials. You will also see that we highlight both the biological and the data or computer science aspects, explaining both the way data is generated and what it represents, as well as the methods and analytical approaches and some basic logic behind some of those methods. So in our practical assignments, you will see that we first review how to do some kind of an analysis and then offer you a chance to practice it on your own, providing you feedback for each one of your submitted assignments. You will also get points for completing those assignments, and those will be limited for a certain period of time. Most of the assignments inside the courses will leverage the big data analysis T-BioInfo platform. Now, this is a resource that is expensive to offer, so we only offer it to the participants of the program, and we are trying to make sure that the resources are used efficiently. In other words, the pipelines that you are going to be using will go from simple to more complex. We'll first try it using the demos, and then eventually give you a lot more guidance on how to prepare a pipeline that is meaningful before you actually start working on an independent project. So once you submit an assignment, that will typically include the result of a pipeline that you might have done some visualization with or done some statistical analysis with. Let me show you how the platform works. The platform is a big data bioinformatics analysis platform designed for biologists and clinicians. It is not intended only for those that plan to be bioinformaticians or are currently bioinformaticians. Our goal is to make these complex tools for big data sets accessible to anyone. Inside the platform, there is a color-coded scheme that you can follow, understanding the logic of why each individual section is being used, and a color-coded number of buttons that connect together, explaining how the pipeline actually works. In the process of building this pipeline, you also get to learn about each step, what it means, and what kinds of input and output data sets are being produced every step of the way. As a result, you will get visual and graphical outputs that help you understand the meaning of the data that you have analyzed. Let me briefly show you how it works. To see the platform, you go to server.t-bio.info. For those of you that have already completed your registration, you will know that you're logged in by seeing your email address right here under this icon. If you still have not received your details, please be patient. You will receive them either today or tomorrow. One of the processing sections that we will use is right here under Transcriptomics, called RNA-seq chip parallel analysis of NGS and microarray data. There are several demo examples that we will review in the next few lessons in this Transcriptomics program. Once you're in, you will see that the buttons that you can use are highlighted in black. By pressing on the button, you learn about each step. For example, this first step will load the data for analysis. Now you have a few options that you can choose from. For example, if we want to map on the genome and understand the top hat algorithm, we can click right here. My next steps have been limited to only the steps that make sense after I process the data using Top Hat 2. For each method, it's important to understand how it varies from other methods and what exactly is the type of analysis that it does. Eventually, as you start building up your pipeline, you learn about the process of transforming the data from raw reads into a table of structured data. In the structured data, you end up with a table called a table of expression. Once the pipeline is complete, you click on End, and you can see the files that the pipeline has produced. These files could be tables, sometimes PDFs, or dynamic links where you can visualize the data. For example, we will learn about individual reads, how those reads are generated by a next-generation sequencing machine, and how they are visualized by mapping them onto a reference genome. We will also learn about how to perform downstream analysis of this data and explore what it means about the individual genes, the bulk data across all of our samples, and how to understand what it might mean in association with a phenotype, for example, a clinical subtype of a cancer patient. In Transcriptomics 2, we will continue to review downstream analysis of such data, thinking about hypothesis testing and regression when we explore the association between a condition or multiple conditions and factors that are included in experimental design. We will learn about complex gene expression patterns and understand how to perform different types of normalization and transformation of our data to make it more appropriate for such hypothesis testing. In Transcriptomics 3, we will turn to machine learning and data mining. So we will discuss how complex data sets like RNA-seq could be used for training machine learning models, exploring different types of patterns in the data and finding groups or making interpretation based on model prediction. We will look at how principal component analysis could be performed using R as well as on the platform, and learn about different types of methods of data visualization, exploration, data mining, and classification in RStudio and in Python. An additional resource that you will have for this program is our code OmicsLogic.com code playground. In the code playground, you will see what happens behind the scenes, how individual buttons that you click are actually coded, and how to manipulate the code so that it is more useful for the type of analysis that you want to do. It provides a cloud-based environment that eliminates the need for installing different libraries and packages, storing all of the files on your computer, and avoids the technical back and forth that is typically required when you're just getting started with code. In the context of this program, we will cover several modules that include how to load data and check for variable data types, how to run descriptive statistics and visualization, do dimensionality reduction with principal component analysis, and do differential gene expression using such packages as DSeq2 and EdgeR. We will also talk about different types of clustering and classification. Let's take a look at an example. For example, when we talk about the differential gene expression analysis, it's important to understand how the data is loaded, what types of transformations are done before we actually perform the statistical analysis, and what do the different outputs actually mean. Once you run the data, you get an editable code that you can modify, understand each section, and run right here in the browser to get the results based on a cloud server that runs the package in R and the data sets that are discussed and explained in the program itself. So such analysis can take a little bit of time, but it's much easier to go through this together in class, understand any challenges, being able to monitor all of the activity together. Once you get the outputs, you can actually save this piece of code and go to your computer, continuing to edit and include it into your Jupyter notebook or some kind of a shared resource that you will include in your final project. Finally, we will start talking about the future of RNA-seq. And it is obvious that when we talk about complex diseases and variation at the subcellular level, we should be looking at how this variation plays out in individual tissues and even individual cells, as opposed to the full organism or the system. To do that, we'll discuss different approaches for single-cell RNA-seq that have been developing since 2009 and today are becoming an exciting resource that allows us to understand and study different types of biomedical projects. So we will talk about different types of methods to produce such data and downstream analysis that also has emerged as several pipelines for processing such data and visualization. Importantly, we will apply all of these tools to basic research and translational research projects that include patient-derived cell lines, xenograft models, and even clinical trials and translational applications for studying how to predict and assign different types of therapeutic interventions. For example, we will focus on the evolution of breast cancer classification and take real publications that offer us an insight into how big companies like Genentech and big research consortia like the Cancer Genome Atlas study such variation and what kinds of conclusions do they come to when they analyze such data. Importantly, after completing this program, you will be able to reach such publications with much greater understanding, learning about how to interpret both the technical and the molecular biology side of each subject that you're covering, and how to apply your understanding and skills to perform similar analysis. So we already heard from Dr. Harpreet Kaur, and now I wanted to introduce you to Dr. Mohit Mazumdar, who is going to be our project mentor for this program, to talk a little bit more about research projects and how you can benefit from research projects in this program. Mohit?
Thank you, Ilya. Good morning, everyone. And yeah, thank you, Ilya, for guiding us through the program. And let me share my screen. Okay, Ilya, it's showing that I cannot share my screen.
Yeah, one sec. Go ahead.
Yeah. So, just a second. Right. So to everyone joining us for the first time, my name is Mohit Mazumdar. I have a PhD in computational biology, and I have been working on various bioinformatics projects in the context of biological research problems to provide bioinformatics solutions, starting from, working from hypothesis to publication. Other than research, I'm developing educational bioinformatics curriculum by collaborating with universities around the globe. So it has been more than a year and a half working with OmicsLogic participants. As you saw, the program, starts with registration and then developing an understanding of NGS data generation, followed by deep diving into the gene expression transcriptomics data analysis, right? For a number of years, our team has been working on project-driven bioinformatics training programs that are designed to help you find, develop, and complete such, research and research projects. So as a result of this experience, we want to offer you several suggestions to make the most out of your research project, whether you're a part of one of the OmicsLogic programs or working independently. There are plenty of opportunities to learn from this resource. Moreover, talking about the portal, on this portal, you'll find like-minded people with about the community that we discussed. So that will help you reflect on your ideas, develop answers to common questions, or you can be presenting to other members of the program and get important feedback from your peers. So using these examples, participants can develop independent or group projects during the program. So many of our past participants had a chance, to present their analysis in front of their classmates or even conference poster sessions and workshops. So just to give you an overview, once you have learned about the NGS data analysis by working with us on various examples and then you're interested, once you're confident and you have learned about the data analysis and you have seen how transcriptomics data is being used in these several of research examples, then you are interested to work on an independent project. So for that, once the OmicsLogic program is finished, or maybe you are learning and you're working on your own project, we have designed a specialized program for our OmicsLogic participants to carry out individual and group projects in collaboration with Pine Biotech and our university partners. So the OmicsLogic Fellowship Program guides researchers to develop and work on NGS data analysis, and that is driven by, it's a bioinformatics project. So the program is structured and starts with, framing the problem. So we start with framing the problem or develop a title. and then identify a data set or data sets and references. So it's important, it's very critical and important stage of the research or the research project is to identify a data set and find those references. So after that, we work on preparing the data, processing the data for analysis, and then perform your analysis, analyze the data in depth using machine learning. And we have already, that Ilya showed you the platform. So we have several machine learning techniques that are implied once you have, have the understanding. So after that, interpretation of the result, so biological interpretation. So that's a program that is structured, and many of our past participants had a chance to present their analysis in front of their classmates, or even, in several conferences, as I said. And one of the examples is Olga. So she is a postdoctoral researcher at Roswell Park Cancer Center, and used her experience, to learn about data analysis techniques. She is now applying in her own, preclinical research. So she's using her knowledge for real research projects, which has data from coming from a hospital. So many others who have continued to benefit from the resources and the training of this OmicsLogic program. So you are a participant, and we expect and we would want you to have a maximum benefit out of these programs. So this starts from the beginners level to faculty and industry professionals. So we have had all those people who have joined us and have equally benefited from these programs. So for those who still have to complete their registration, we would welcome you to join this program and earn a certificate of excellence for advanced projects or a certificate of participation for basic training. So the sessions will guide you, will help you, and the additional resources that you saw. And then you have support, you have the trainers, you have the mentors. So let me now pass it to Ilya for maybe conclusive remarks and the rest of the session. So if you have any question, please reach out to us. My email address, I'll just put it in the chat, mohit@pine.bio, and we can communicate through the forum as well. So that is there. So really happy to have you all.
Mohit.
Yeah, thank you, Ilya.
So I think that this program is going to allow all of the participants to both explore the basics of RNA-seq data analysis, understand how it applies to several different examples, practice those tools and develop some basic skills, and work with our team to develop and conduct your research in the course of this program, which is about 1 month, or right after that by joining a mentor-guided research fellowship, as Mohit just explained. In the program itself, you will have access to projects as well. These include several publications that we have went through and converted into educational data sets that you can work on and understand all of the different aspects that you will have to have in your mind as you analyze and interpret that data. As Mohit mentioned, the outcome of that process is being able to present a poster or a presentation in front of our peers in the program, and potentially continue on to present this at different types of conferences. Right now is a great opportunity to join interactive online poster presentations at a variety of poster sessions around the world. Our mentors that you have seen in this program and the interns in our intern and research fellowship programs are here to continue and support your work. And we're going to be working with you in the second part of the program to make sure that the projects are both impactful and satisfying to you, whatever your objective is, whether it is developing your career, making sure that you are ready to join a specific research lab, or even just for your personal research as a PI or a researcher in a lab. So as Mohit mentioned, in the course of this program, you will learn RNA-seq data processing, analysis, and interpretation. You will be able to leverage cloud-based tools for hands-on assignments, apply machine learning to large biomedical datasets, complete and present independent research projects, and obtain a certificate of excellence and recognition based on your achievements in this program. Thank you for joining today's session. I encourage everybody to just make sure before we leave, for those of you that have completed the registration, by going onto the home page for the OmicsLogic Transcriptomics program. Make sure that your profile is complete, as Beepsa mentioned, and that you start reviewing some of the resources that she highlighted in the logistics part of this presentation. You will also find our individual profiles, and you can reach out directly to us or post any questions you might have on the forum. And we are excited to see you in our next session. You can learn about the schedule of these interactive sessions and associated courses by going to the syllabus tab. And on the syllabus tab, you will see that today, this was our program overview session. We ask everybody to start with going through the Introduction to Bioinformatics course. Now, the reason for going through that course is because it also includes an important survey that will help us understand more about you and more about your background. So we are here to talk to you about what we know about RNA-seq, but we're also extremely interested to learn more about you, which includes your research interests. So we want to understand what you are interested in. We're also interested to understand your level of education. So what is your background? What is your technical background? If you know anything about bioinformatics, or is this completely new to you? If you have done some research using RNA-seq data, or you have not. And also to see how we can best help you throughout this program. So if you go to the syllabus tab, and Beepsa, if you don't mind putting the link in the chat there as well, you will see that after this first session, we actually will ask you to complete this Introduction to Bioinformatics course. And in our next session, we will pick up from here and continue talking about going through the next generation sequencing processing steps and what is the meaning of every single step, and why would we want to take one option or the other. For those of you that will be continuing with this program, that have completed your registration, please add marketing@pine.bio to your contact list. You will be receiving emails with links to next sessions. They're going to be different from this Zoom link. You will also get important announcements about the program and additional resources that we will be using in the sessions themselves. So thank you again for joining today. We are excited for this program to start, and we will be looking forward to seeing you in our next session. And now I'll just open this for questions if anyone has any questions. So let's take the first one. Parisa is asking, I'm working on fungal and bacterial genome. Do we have any courses related to DNA-seq analysis of those creatures? So we just completed a metagenomics program that does look at different types of bacterial genomes. And so we encourage you to take a look at Introduction to Metagenomics, Metagenomics 1 and 2. We also have a project that deals with some viral sequencing data. So yes, we'll be happy to share that. Please leave your email so that we can communicate with you by email and send you these links. Okay, about any logistics or any kinds of questions that you might have about the program and joining the program, please email marketing@pine.bio. For the code, what code will be saved and how can we re-access the same? So all of the code is actually going to stay accessible. So you can see right here, my code has finished. I can see the outputs, but also I can save the code. Now, similarly to this, in the pipelines that I showed you on the server big data platform, you will have a number of different methods that you can run and then save the code onto your computer for how that code was run with your input files and with all of the settings. So all of this material is something that you just store on your computer and directly open in your RStudio or any kind of Python IDE. And obviously, once your environment is set up and you've got all the same packages, it should run exactly the same on your computer. Will the projects that we will be doing in this program be suitable for publication? So whenever we deal with real research projects, it's important to remember, and Harpreet and Mohit can probably add to this, it takes a while, right, to collect the data, to analyze and explore the data, eventually coming to a real kind of a research level, publication level project. So typically we see that that takes anywhere between 3 to 12 months. So that's why in the context of this program, the project that you're likely to complete is going to be one of your first steps. So you're going to just develop a sense of what you're interested in, develop a project title, explore the data, make some preliminary analysis, and potentially have some interesting results that you can then continue to develop into a real full research proposal. Because it takes a longer time, and it also requires many different types of assistance that are very specific to that project. Instead of a program that has a detailed plan, it's a kind of a rolling basis. You can enroll into the research fellowship to benefit from the tools, the mentorship, and the support that we offer through this program. Do we need specific notebook on my computer system so that saved code can be re-accessed? Well, all code, if I save it, it actually you can open it in any text editor. It's just code. To run it, however, you probably will benefit from installing an environment, for example, RStudio or just the R packages that will be used. In Python, it's the same way. You can run Python scripts from the terminal, but you will have to install Python and install different packages. There are also developer environments that are integrated and provide you with some environment where you can easily access the files, the outputs, the code itself, kind of learn the syntax, etc. So we will review some of those aspects in the program as well. Which language is better, Python or R? There's a debate about which one is more popular. You know, better needs to be defined more, I think. You know, are we talking about efficiency? Are we talking about ease of use? Are we talking about how they handle, you know, different types of data sets, or maybe what packages are available? So I think, you know, that's an open discussion. Do I need to be affiliated with some university or do a project for a research fellowship? No, you don't need to be a part of a university. We have many independent participants that join that program as well. You know, the university provides you with an environment, and that's what our program seeks to provide as well. In fact, Beepsa, if you could share links to our social media groups on LinkedIn and on Facebook. We have groups with several hundred of participants that have either completed or participated in some way in our OmicsLogic programs. You will see that they include different levels of students at different universities, researchers, as well as faculty and industry professionals. So we are continuously building a community of participants that can help each other by providing feedback, potentially collaboration, and there's a lot of opportunities to for employment, for research in different labs, etc. Basic R or Python that could be used in genomic data science. Yes, in the context of this program, you will start from the very basics. So in fact, one of the first lessons that we will go through is how to load and check for variables in both R and Python. So I think you're going to be starting from the very basics. If you don't know any coding at all, you can join this program to learn both the coding and the application to RNA-seq data. We will have another program that is called OmicsLogic Data Science. That program will start in October, November. And so you can benefit from just more of a data science perspective, a much more detailed review of the different coding practices, different data science applications, all related to genomic or biomedical data sets. And Ankit, if you want to share your email with us, we will be happy to email you the details of that program as well. Okay, are there any other questions? Okay, excellent. Thank you for joining today again. Those are some of our social media groups. Thank you, Mohit, for sharing those links. You can join the LinkedIn group. We have a wide variety of professionals from the industry, from academia, previous participants of these programs. You can learn about how they benefit from this program and what are the outcomes. Some of them got hired, some of them went on to do their research, etc. So you will benefit from interacting with those participants as well. Do you need to read anything to prepare for tomorrow's session? Yes, please go through the Introduction to Bioinformatics course. So we will definitely include you. Again, if you have any questions about assignments for the next class, you should be able to see them on this page right here. So you see that for today's session, the assignment is to complete the Introduction to Bioinformatics course. As I mentioned, the Introduction to Bioinformatics is an overview of how bioinformatics is applied to a variety of different types of challenges in the life sciences in general, including agriculture, biomedicine, and others. And one of the first lessons is going to be a survey that we ask all of you to fill out. In this survey, we are looking to learn about you so that we understand how we can best organize the materials and make the program effective for you. So if you could finish this course, you will also learn about how this resource will work throughout the program. Okay, so thank you again, all of you, for joining today. Please share this program with your contacts. We're looking forward to building a long-term relationship that we hope will be beneficial to all of us. And we want to, of course, see you in our next session and continue this conversation. Thank you, everyone.